The title of Inventions about a novel drug delivery system based on magnetic, composite, and organic materials for transporting cisplatin to cancer cells. Table of Contents: Introduction, Methods, Main Result, and Conclusion. Number 1. Introduction. Cancer is an uncontrollable and growing disease recognized and as one of the leading causes of death worldwide. The liver is the largest and most complex organ in the body, and the liver cancer ranks 6th among the most common cancers and 4th among the deadliest cancers globally. Finding optimized methods for cancer treatments is circle. One such method is using drug carrier components to deliver drug to the targeted tissue. In this invention, a nano composite of iron, cobalt, zinc, along with chitosan nanoparticles extracted from shrimp shells with high zeta potential and silver nanoparticle synthesized through green chemistry from elderberry extract are used for the delivery of the drug Cisplatin to liver cancer cells. The result showed that this system can serve as an effective carrier for delivering the drug to the target tissue, which in this case is liver cancer cells. Number 1. Introduction. The effectiveness of many drugs often depends on their ability to reach the site that requires treatment. In many cases, only a small amount of the administered drug reaches the target tissue, while the majority of the drug, along with its chemical, physical, and biochemical properties, spreads throughout the body. One of the most critical issues in drug delivery is controlled drug delivery to the body. In conventional drug consumption methods such as oral or injectable, the drug is distributed throughout the body, affecting the entire system and causing side effects. To achieve a specific therapeutic effect, large quantities of the drug are required. Therefore, developing a drug delivery system that enhances drug activity is crucial. The idea of using submicron drug delivery system for targeted drug delivery became visible after Paul Ehrlich proposed the concept of magic bolt drug carriers, charged nano or microparticles, particularly those combined with biodegradable polymers bound to specific drugs, have a unique capacity for drug retention. In the design and development of drug delivery carriers, the goal is to achieve a system with suitable drug loading capacity, desirable release properties, prolonged half-life, and low toxicity. Common carriers used in drug delivery included missiles, liposomes, nanoparticles, dendrimers, liquid crystals, hydrogels, and conjugates. Among these drug-carrying nanoparticles exist in the forms of nanocapsules and nanospheres. This system can absorb and encapsulate drugs, thereby protecting them from enzymatic and chemical degradation. Nanocapsules are hollow systems in which the drug is confined within cavities surrounded by a polymeric membrane. Nanoparticles made from macromolecules can act as drug carriers capable of dissolving, interpreting, encapsulating, absorbing, or chemically binding the drug. The polymers used to prepare nanoparticles can be either natural or synthetic. One method to improve targeted drug release involves using an external magnetic field to guide the drug to the desired site. In this method, the drug is attached to magnetic nanoparticles with an appropriate coating and then injected into the circulatory system. A magnetic field is then applied to the target area, selectively directing the magnetic carriers to that site. Once the magnetic field is applied, the particles are trapped in the target region, allowing the drug to directly release at the intended site. Number 2. Methods. Carriers based on charged particles with positive charge possess significant potential for targeted drug delivery. Additionally, nanoparticles delivered from medicinal plant extract and biopolymers are widely used as carrier agent for delivering drug to host tissues. In this invention, silver nanoparticles extracted from the elderberry plant were utilized due to their tunable size and shape, ability to enhance the stability of nucleic acid attached to their surface, high density land surface attachment, exceptional membrane delivery capacity, protection of bound drugs from degradation, and potential to improve intracellular drug delivery in a highly controlled manner. Furthermore, chosen nanoparticles delivered from shrimp shells with high zeta potential and medium molecular weight were employed to increase drug retention time by preventing its diffusion and improving adhesion efficiency to host tissue. Iron, zinc, cobalt nanocomposites were also incorporated to accelerate drug deposition at the target site in minimal time, achieving efficient targeted delivery for liver cancer cells. The drug carrier developed in this invention for delivering drugs to liver cancer cells consists of iron, zinc, cobalt, nanocomposites, chitosan with a high degree of detaglation and high zeta potential, and silver nanoparticles delivered from the aqueous extract of elderberry plant leaves. The method for synthesizing these novel carriers is as follows. A 200 ppm colloid of silver nanoparticles was prepared from elderberry plant extract. Number 2, a chitosan solution 0.1 g in 0.5% acetic acid with high zeta potential and high distillation degree was prepared, and an iron zinc cobalt composite was chemically synthesized. Number 3, 2 milliliters of each solution were taken, mixed, and disappeared. Number 4, the final composite solution was functionalized with 10.8% V/V orthosilicate at 70 degrees to activate functional groups, and the drug was loaded onto the composite. Number 5, the final reaction was performed using polyethylene glycol, and the carrier with the drug was tested on liver cancer cells. The final product can be provided as a powder to research laboratories for use as a drug carrier. I want to talk about the charts. Figure 1, time killing of nanocomposite and the nanodrug Cisplatin. MIC result, antibiotic ipsine 256 microgram milli, nanocomposite 64 microgram milli, nanocomposite with drug 64 microgram milli. MBC result, antibiotic ipsine 512 microgram milli, nanocomposite 128 microgram milli, nanocomposite with drug 128 microgram milli. Image depicting the bacterial killing time of the E. coli under the influence of the composite combined with the drug, as well as the MIC minimum inhibitory concentration and MBC minimum bacterial concentration of the bacteria. Figure 2, visibility of HEPG2 cell after treatment by nanocomposite and drug Cisplatin, showing the carrier with the drug on liver cancer cells. Figure 3, visibility of HPG2 cell after treatment by nanocomposite, showing the carrier without the drug Cisplatin on liver cancer cells. Figure 4, visibility of L929 cell after treatment by nanocomposite and drug Cisplatin, showing the carrier with the drug Cisplatin on liver healthy cells. Figure 5, visibility of L929 cell after treatment by nanocomposite, showing the carrier without the drug Cisplatin on liver healthy cells. Figure number 4, microscopic image of liver cancer cells and healthy cells in the presence of the carrier with and without the drug. Table of content number 3, conclusion. Advantages of the claimed invention compared to prior invention. Number 1, development of a novel nanocarrier for testing various drugs on liver cancer cells in laboratory setting. Number 2, a highly efficient drug carrier composition for delivering drug to liver cancer cells. Number 3, acceleration of drug testing on cancer cell growth in the shortest possible time. Number 4, prevention of drug diffusion to other cells, ensuring targeted drug delivery to host tissues. Primary application of the invention as described. The primary application of this invention is in research laboratories, particularly in centers focused on various types of cancer. It falls within the field of molecular biology and cancer therapy. The invention is suitable for use in drug synthetic and testing on cancer cells, research laboratories working on cancer, pharmaceutical development centers.